Hello, my name is Claire Cook and I'm a graduate student at the University of Michigan, and today I'm excited to talk to you about epigenetics. So epigenetics is probably most evident if we consider our organs. Let's say the brain and the liver. The brain is responsible for processing sensory information, whereas the liver is responsible for processing nutrients from the foods that you eat. These are fairly different roles, so it probably comes to no surprise that if you look closer, their cells look very different. They have different structures and different molecules that they use to function. But it might come as a surprise that if you zoom in even closer, they have the same DNA. Now, DNA is like a blueprint for those structures and molecules that these cells use to function. So how is it that two different cells would have the same blueprint? The explanation is that they actually use different parts of the DNA blueprint. And epigenetics is what controls what parts of the DNA are used in different cells. Epigenetics is like an interpreter for DNA, and it translates the information coded in a DNA molecule into characteristics that you and I can see. It does this using a variety of tools. Some make the DNA more open and available to be used, whereas others actually block the DNA and prevent it from being used. And scientists are really interested in how cells use the epigenetics toolbox in response to different conditions and different environmental cues. They use tests that give a snapshot of where these tools are being used along the DNA molecule and how that translates into what parts of the DNA can be used. Unfortunately, these tests commonly look at millions of cells at a time and merge together all of the responses from those millions of cells. This is similar to envisioning a picture of a mountain. There are pixels in this picture that make up the different parts: the sky, the mountain, the clouds. But if you mix together all of these pixels, all you see is gray, and you don't see any of the distinct parts of the picture. And that's basically what these tests do. Ideally, these tests would give a good representation of all of the cells present in our sample, but in reality, all these cells use the epigenetics toolbox in slightly different ways, and these tests don't give any insight into those differences. This is really important in two cases. The first is that these tests can actually overlook smaller responses from rarer groups of cells. And the second is that these tests can actually merge together the responses from two distinct but equal groups of cells. And scientists are trying to improve these tests so that we can get more accurate information about the cells in our samples. And one of the ways they're doing that is by using lab-on-a-chip research. So in lab-on-a-chip research, scientists are trying to take all the steps included in an epigenetics test that are typically performed on a lab bench and to miniaturize them so that they can all fit on a device that you can hold with one finger. And the benefit of this miniaturization is that we don't need as much sample to get similar results. So we can envision, slowly but surely, scaling down our sample size until eventually we're looking at a single cell and getting accurate information about how that single cell uses the epigenetics toolbox to be able to function. And we can do this for many cells and begin building back up to a more complex system and getting a better understanding of how all the cells within this complex system use the epigenetics toolbox in slightly different ways to be able to work together.